This is the microbiology laboratory. In the microbiology laboratory, we detect microorganisms associated with causing disease, such as bacteria, fungi, viruses, and parasites. We also would determine treatment options, and this is done largely by antimicrobial susceptibility testing. In terms of the samples we would receive a various range of sample types, and I'm going to be showing you some of those as we move along. In this section, we would process a number of swabs, fluids, and tissues. Swabs would be received up in containers like this. So the swabs will have been taken, placed into the container. There's fluid at the bottom, and any bacteria present will be in the bottom of the container. The samples are then added to this rack, where they'll be then processed on our walk-away specimen processor, which I'll show you now. This is our walk-away specimen processor. It's called the Previ-Isola. And how it works is we load up all the media that we require for each particular sample type onto the left-hand side. Then the samples that we want processed, we just put into this compartment. Check that the correct plates have been loaded on the left hand side, and then start. The fluid from the bottom of the container is aspirated onto each appropriate media type, and this labeler will label each individual plate with the accession number and patient details. And once the specimen's been processed, we will get it out here in this output cassette, which I'll be able to show you an example in a minute. The samples have been processed. The plates which have been inoculated with the fluid from the swabs, they come out into these output cassettes. So you'll be able to see here how they're labelled up. So they're labelled up with the accession number as well as the type of swab that was initially put onto the system. So these plates here are typical plates we would set up for a wound swab. This is a culture plate after 24 hours incubation. You can see the bacterial colonies growing the whole way around the plate. But what's most useful to us are these individual colonies at the end, where we can do further work on and help determine the antibiotic of choice for the patient.
We're in our epidemiology section now, so this is where we screen for MRSA, and we do our neonatal screens here as well. All the little babies and neonatal units get checked out here. Okay, so we would receive upwards of between 50 swabs per day in this section, and basically what we're doing is screening to make sure there's no MRSA present, and the result goes out then within 12 to 18 hours. However, if there is MRSA present, then a whole gamut of control of infection swings in then and be reported as such. Now, a negative sample is going to look something like this, and thankfully most of these are. However, when we do detect an MRSA, we use chromogenic agar in this section, and if there's MRSA present, it looks like that. You see? So obviously if we see something like this, we report it straight away to our consultants and our control infection team, and then they notify the wards and act appropriately. We're in our urine section now. So every single day we receive between 250 and 300 samples per day, and each one of those is looked at directly through microscopy, and we will culture every single urine as well. Okay? So pretty much what happens is every sample in its microscopy, when it passes through the machine, it takes 10 snapshots and brings up the pictures on the screen. This result is then transferred to our computer, and then we look at the direct result along with what we have grown the next day to make our analysis on whether it's an infection or not. If it is a negative sample, then we get a nice plate like this, which is all no growth. There's 6 samples in this plate, and every one of them is negative. If, however, there is a urinary tract infection present, we'll see something like this. Now, if I show you this side of the plate, you can see there's 3 organisms growing on 3 different patients there. So it's our job then to sub that out and identify what antibiotics are most appropriate. An antibiotic plate would look something like this. So I can show it that way. You can see in this plate 3 antibiotics are useful and 3 are not in this case.
This is the Category 3 laboratory. In the Category 3 laboratory, we process feces and sputums. In terms of the sputum samples or respiratory samples, we use these to help determine any cause of lower respiratory tract infection. A couple of examples of bacteria that we may isolate from a sputum sample are Haemophilus influenza or Streptococcus pneumonia. In terms of feces, there are a number of examinations carried out there as well, examining for enteric infection and examples of bacteria that we may isolate. As you've probably heard of some of these, Salmonella, Campylobacter and C. difficile. We'll also look for parasites. So if a patient has been away on foreign travel to some exotic country, they may have brought home a parasite. So again, we'll examine that in here. All work is carried out here in Category 3, so we may deal with high-risk specimens. So we could have a risk of TB from the sputum samples, so everything has to be carried out in here. We wear blue-coloured coats because we don't want to bring anything that we may have been working with out into the rest of the laboratory. So it's very much protecting ourselves and protecting everyone else as well.